So this is the PowerPoint for Chapter 12, and this is actually Part 1. And the first thing as we're getting started is to recognize that the ability of an organism to reproduce best distinguishes a living thing from a non-living thing. So this chapter is going to discuss the cell cycle, cell growth, and cell division. So in unicellular organisms, division of one cell reproduces the entire organism. But when you have multicellular organisms, cell division is going to be used for reproduction, for growth, or for repair and renewal. To quickly review some of the structures of the cell that are involved in cell division, we start with the nucleus, which again contains chromosomes and DNA. And then we also have to include the cytoskeleton, which has a key role in cell division. In particular, the centrioles, which are part of the cytoskeleton. And also the microtubule spindle fibers that make up the spindle that pulls apart chromosomes during cell division. Reviewing the nucleus's function is to protect the DNA. And an important part of its structure includes the pores that are in the nuclear envelope. So again, the nuclear envelope is a double membrane, and those pores are important because of both things that need to come into the nucleus, such as signaling molecules, signaling the beginning of either replication of the DNA or transcription factors that are involved in later the actual production of proteins from the DNA. So those pores need to be large enough to allow the signaling molecules to come into the nucleus. And you can see here the size on the right there, it is actually pretty large. The cytoskeleton, its major function is actual structural support of the cell. It also plays a part in the movement of the cell, its motility. So both in the way that we're learning that the cytoskeleton can change within a cell, allowing the cell to move. And also on the outside of the cell, the formation of cilia or flagella that actually can move a cell. And the cytoskeleton is also involved in the regulation or cellular regulation by organizing structures and activities of the cell, and in particular during cell division. And this is just a cross-section of a cell showing its membrane, different organelles, and then it's also showing you parts of the cytoskeleton and how they provide a structure for the different organelles to, have their place within the cell. The centrioles, which are part of that cytoskeleton, are a pair of microtubules that actually have a very specific shape, as you can see there in the lower right-hand corner. And they're used to guide the chromosomes as they move about in the cell during mitosis. So what exactly gets passed on during mitosis? What do daughter cells get? And essentially what they get is an exact copy of the genetic material, or the DNA. That DNA, it's important to remember what is DNA. So DNA is actually all of the DNA together makes up the cell's genome, or actually we would even say the person or organism's genome consists of the DNA. And that genome can be a single DNA molecule, or it can be a number of DNA molecules, which is more common in eukaryotic cells. And the DNA molecules in a cell get packaged into what we call chromosomes. And those chromosomes consist of chromatin, which is a mixture or a complex of DNA and protein together, which is what condenses and gets duplicated during cell division. The other thing that's passed on to daughter cells are organelles, cytoplasm, cell membrane, and enzymes. And those things become part of the daughter cells as cytokinesis occurs in mitosis. So where we all begin is at a very small cell. It's actually smaller than the period at the end of a sentence. You can see the red arrow is pointing to a period there. And then from that cell, which is the fertilized egg, mitosis occurs. And mitosis occurs and occurs over and over again, resulting in the complex organism that we become. And mitosis, we divide into different phases, as you guys, I'm sure, will remember. An acronym to remember is IPMAT, which stands for interphase, prophase, metaphase, anaphase, and telophase. But we also want to add in a couple extra steps. Right after prophase, we have prometaphase, which is an in-between step before we get to metaphase. And also at the end of telophase, we have the actual cytokinesis occurring, which is the actual division of the two daughter cells. But before we look carefully at mitosis, we should look at the bigger picture, because mitosis only accounts for about 10% of the cell's life cycle. The majority of the life cycle of a cell is spent in what we call interphase. So interphase is basically what the cell is doing its everyday job. So during interphase, we have things like the production of RNA, the synthesis of proteins and enzymes occurring. What that cell's specialization is in terms of its function within the organism. During interphase, we also have a portion of that time which is involved in preparing for duplication of the genome in order for mitosis to occur. So the mitotic phase is what we call the M phase. In interphase is the cell growth and copying of chromosomes in preparation for cell division. Interphase can be divided into 3 subphases. This isn't a very good slide, but you can still see the cycle there. Those 3 divisions are the G1 phase, or the first gap, or gap 1 phase. There's the S phase, which stands for synthesis phase, and G2, or the second gap phase. And cell growth is actually occurring through all 3 of these phases. But the only phase that the chromosomes get duplicated within is the S phase. So synthesis is really referring to the duplication of chromosomes. Now what this is going to show here is that depending on the type of cell, the life cycle is going to actually vary within the cell. So when we talk about brain or nerve cells or muscle cells, they actually grow and mature, but they never divide again. So there's no reproduction of those cells. There's no duplication of those cells. We don't get more of them. So what you have once development has ended is the number that you're going to end up with for the rest of your life. So that means the cell cycle never goes into that M phase. And so it actually never needs to go through the S phase or the G2. So it really only goes through G1, an initial growth phase, and then it spins off into what we call a G0 phase, which is a resting phase. And the cell is not actually at rest. It's doing its work. It's doing its protein synthesis job. But it is never going to go into that S or G2 phase. In contrast, all the rest of the cells, like epithelial, blood cells, stem cells, every cell, liver cells, they actually do go through all the different phases because they do divide. And so we do get new and fresh epithelial cells or new and fresh blood cells. And that occurs through those 4 phases of the cell cycle. So this slide is actually just showing that we have, and sorry about the sound effects, but this is showing the 3 different phases that we have, again, through interphase. So what it's showing here is just, again, the G1 or first gap. And gap you can also think of as the first growth phase. So this is when the cell is doing its job. Its focus is on that protein synthesis. And then we have a signal occur where the S phase then begins. And this is when DNA is synthesized by the copying of chromosomes. This is when we have replication of the DNA. And then that will bring the cell into the next phase, which is the second gap or second growth phase. So there is more cell growth that occurs here. And this is in preparation for division. This is also the phase when organelles are produced, more proteins, membranes are being produced. The things that are going to be needed for the actual division into two daughter cells. So during interphase, if you were to look at a cell, you could see that the nucleus is very well defined and the DNA is loosely packed in long chromatin fibers. So the graphic depiction there on the lower right-hand side shows you those loosely packed DNA. In preparation for mitosis, the replication of chromosomes will occur. Oops, let's go back here. And again, the proteins and organelles are created that are going to be needed for the daughter cells. This picture on the lower right also is a nice graphic to show you the centrosomes with the centriole pairs up there in the corner. So you can see how those centrioles are actually together when the cell is in its interphase. The replication of DNA should be something that's familiar. This is occurring during that S phase or synthesis phase. It's when the cell is going to make a copy of its DNA. And going back again, I just want to make sure you have a chance to look over these different facts. The human cell duplicates about 3 meters of DNA. And with all of that replication, there is still only an error rate of about 1 per 100 million bases. So essentially, if you do the math with 3 billion base pairs in a mammalian genome, that's only 30 errors per cell cycle. But it still is 30 mutations or 30 different errors that are going to occur and give variety actually to the next daughter cell's genome.